Hi, good afternoon, everybody. It's a pleasure to be here. And so I'm a computer scientist. My field is medical imaging processing, but we are a pathology lab. And we work especially with dementia. And we work especially with Alzheimer's disease. So I'm going to talk today about my role in this lab. So why we are interested in Alzheimer's? First is because it's one of the main reasons of death in the US. And it's the only disease that has increased in frequency in the last decade. And currently, there's about 5 million people living with Alzheimer's. And it costs more than $200 billion a year in health care. We estimate that by 2015, there will be 40 million people, which means it's like about 1 every 8 people who have Alzheimer's will be living with Alzheimer's, and it's going to cost about $1.1 trillion. And Alzheimer's can affect everybody. It's just not selective. And it's progressive disease. So it's characterized by neuron death and tau protein tangles. And these two factors, they're closely correlated to the cognitive decline that we see during the disease progress. And it starts silently in small regions of the brain. And it spreads as the disease progresses and goes to the limbic system and finally to the whole cortex. The big problem with Alzheimer's is that the biology underlying this is not really well understood. For instance, we don't know if those tau tangles, they really cause the neuronal death that we're seeing. And we know that disease begins many years before the actual symptoms kick in. So the best time for treatment would be starting while the disease is still silent. And in our lab, we are interested in tiny regions of the brain, the brainstem, called locus ceruleus, dorsal rough, and substantia nigra, because in 2009, we showed that these regions are the first affected by Alzheimer's when the disease is still silent. And also some of interesting things, like unlike the hippocampus, which changes even in normal aging, the volume changes over normal aging, LC doesn't change as people age. So it could be a nice biomarker. So, and here is the brainstem, for those who are not used to brain anatomy like me. And these tiny regions, they're located inside this brainstem. They are some millimeters wide, so they're very tiny. In red here, we have the dorsal rough, and blue here is the LC, locus ceruleus, and green here would be the substantia nigra. Problem is, we work with pathology, which means post-mortem tissue. We are trying to understand the disease from this kind of material, but we want to be able to translate our findings to treatment of living people. And the best way to image brain in living people is MRI nowadays. Problem is, we can't really see these regions in MRI because they are so tiny that current standard MRI acquisitions don't resolve them. So what do we want to do? You want to understand how the disease is working, is progressing in a microscopic level in these tiny regions. And for this, we need to quantify cells. We need to quantify the amount of protein that we have in these regions, the tau protein. We need to quantify the amount of cell death that we have in these regions. But we want also to be able to see these regions, spot these regions in MRI, so that we can translate these findings in microscopy to MRI signal, so we can correlate these findings with MRI signal. And today, I'm going to talk about 2 projects that are working parallel in the lab. And one is image registration, which is structural microscopic view of the brain. And the second one is using machine learning for counting the cells, detecting the cells in these images, and so that we can bring up more productive analyzing microscopy, so that you can have a more reproducible result. And the first problem, which is the high resolution registration. High resolution here, I mean, it's higher than the usual registration resolution that we see for registration studies, but it's not as high as microscopy. And so what kind of material do we have? First, we have a brain that we get from patients that enrolled in one of the projects in the lab. And this brain is scanned while still in the patient's head, and we have a standard clinical MRI here. And then this brain is extracted. It goes through a series of chemical process to be fixated. Sounds like zombie stuff, I know. And then this brain is embedded in what's called celluloid gene. And when the block's ready, we put this in this equipment called the microtome. And over this microtome, we have a camera fixed in a way that it cannot move during the slicing process. So before every microtome stroke, we take a picture that we call block face. After all the brains is sliced, these slices go through a staining process with glycine, and they're mounted on glass slides, and they're again imaged. So we end up with 3 volumes: the MRI, the block face, and the histology. And the histology is also imaged in a way. We don't use a microscope here. We use a Canon camera with a macro lens, which is enough, just enough to give some resolution on, for instance, cortical layers. But it's not as high. We don't have such a high resolution as a microscopic image. And the idea that what we want here is be able to align this histology to this MRI. Problem is, during all the chemical treatment and slicing, we create a lot of artifacts. First, when the brain is extracted from the skull, the brain has a very soft tissue and it gets squashed. And after fixation, it shrinks. And we have a lot of nonlinear and linear deformations all over the brain. We also have a lot of tears, shearing, and we lose the 3D shape when we do the slicing. And also, we don't have the same slicing plane that we have when we do the MRI excision. So we never know which histology slice corresponds to which MRI slice. We cannot just do this direct comparison. Moreover, we have a lot of data. Even though this is not a microscopy data set, we have a lot of data. Our histology, our original size, the original resolution of this histology is 0.02 by 0.02 millimeters in plane. And it yields about 80 gigabytes. One brain has about 300 slices, 400 slices, depends on the size of the brain. And it's going to yield about 80 gigabytes of data if I work with double precision, which is the usual type of data that registration algorithms use. And 80 gigabytes if I'm working grayscale. If I need the color, which is our case, it's going to be 3 times more. So in the beginning, people were trying to do this manually, literally segmenting out the images using Photoshop, stacking them manually, trying to align them using a software called Amida, and stretching them, doing all kinds of stuff, but it wasn't really effective. So my role in the lab was come up with a pipeline that would help automate this process. And basically what we do is use the block face as a template for realigning the histologies, creating an intermediate volume, and then registering the volume to the histology. And I'm going to talk about the steps that we need to get this registration done. First is going to be a segmentation step. We have some pre-processing, and the segmentation is responsible for getting rid of all this cluttered black background that is not important to us, and it's going to cause error if you try to register those images, one image to the other directly without using any kind of landmarks. So what I do is I model the problem as a Gaussian mixture model. First, I convert my RGB images to the YIQ space, because we notice that in this color space, our tissue pixels, they tend to cluster to a specific region in the space. So it's easier to model the problem. And We use Gaussian mixture model, and we compute the model parameters using expectation maximization. And this way I come up with a first mask that I can refine to segment the brain region. I do the refinement using active contours. And this is an example of the results that we get. And those are the original images, and these are the segmented images. And my next step is that for each slice, I need to align the histology, which is this one down here, to its respective block face. And I am using the block face. So when I slice the brain, I lose this 3D shape. If I just stack it back, I'm going to end up with a squashed, twisted brain. Then that's not what you want. And the block face, although it has been squashed after the surgery, but you can still recover the 3D shape from this block face image. And as we are careful and you don't allow the camera to move, we have these slices very well aligned, and we can use them for locating the histology. So I do for each slice the registration using a fine registration algorithm. And after the registration, I save all my matrices for future use. And this is an example of the registration. Here, in pseudocolor is the histology. I use pseudocolor just for contrast purpose. And this is the registered result. I can see, I don't know if it's possible in the projector, but you can see that the boundaries are aligned in this checkerboard view. So after I do all this 2D alignment, I need to stack my images. But before I do that, I have to correct it for intense inhomogeneity. Because what happens is that even though we are very careful during the staining process, we are very careful during the image process, careful with lighting conditions, staining time, we always end up with slides that have slightly different intensities. So if I just stack them, I end up with these stripes here. And they're not good for visualization. They're not good for registration. So we try to minimize this problem as much as we can. And we do this by using an iterative method where I pick my middle slice as the reference, and I compute the continuous histogram of this slice, and I compute the continuous histogram for my neighbors above and below this slice. Then I match both histograms, so it's a global procedure, using a fine transform, and I compute these parameters using the Paulo optimization. Then I just remap the colors using the new histograms, and I get the reference, my next neighbor as the reference, and do this in an iterative way for both halves of the volume. And I will end up with a volume which is much better than the other one. It's not perfect. You still see some of the stripes, but it has a lot of improvement. And just out of curiosity here, this is a 3D reconstruction of the intermediate volume surface. And for people who've seen a brain, they can tell that this is squashed. And just out of curiosity, you can see like cut here. This was caused by the knife when they were doing the surgery. So after we do this pre-processing, we have to resample histology, which used to be 0.02 by 0.02 millimeters. And why we do that? I like to have my histology as high with the most resolution that I can have, because I can see the inner brain structures that I'm interested. But I have to do this resampling to reduce memory footprint. Why? Because most registration tools that we have nowadays, they're not ready for big data. They don't work well with big data sets. They don't scale well. And in our case, 0.15 cubic millimeters was the highest resolution we could use without running out of memory in our computer nodes. This was ran in NERSC computers on the LBL, but still, you were running out of memory because this algorithm doesn't scale. So I did this resampling using cubic spline, and in parallel, I was also pre-processing the MRI to get rid of the main artifacts, which the first one is the field inhomogeneity. And for those who have worked with MRI, probably have seen this, is you have this variation of the static field in the MRI, it always causes this kind of slowly varying intensity problem, which can be a problem for segmentation, skull segmentation, for methods that rely on intensity and contrast. So I use an algorithm called N3 for correct this. And here is like the original, this is the corrected, and this is the field that was computed from this volume. So after that, I segment the brain, because again, I need to get rid of all of these uninteresting structures that just cause error in my registration. And I also resample my MRI. Why? It's a 1 cubic millimeter volume, and I resample this to beat the histology resolution, which is 0.15. Why I do that? I'm not getting any new information from that, right? Yeah, but if I don't do this, the registration algorithm is going to bring down my resolution even more for the registration. So I need to have my target volume with the same resolution that my histology volume. And that's why I'm doing this resampling right now. And finally, for the 3D registration, I have my histology. I have to center my histology to MRI. And this is still a manual step in our pipeline. Hopefully, we'll be able to automate this in the future. But what happens is that when I do the scanning MRI, the scanner computes for me coordinate matrix that tells me how my volume is oriented in space. But my histology doesn't have this matrix because I did it manually, build my volume. So I do a manual registration, from which I use FreeView as the tool FreeView, from which I can compute and find a matrix and center both volumes. Finally, I use the diffeomorphic registration method, which is called symmetric normalization. It comes in a toolbox called ANTS. And it's a really good method for brain image registration. The fact that it's diffeomorphic is important for us because it guarantees that the mapping is smooth. So I don't get foldings in my tissue, and which I also don't get disappearing pixels because it guarantees to me that I have a 1-to-1 mapping. I don't want to. Why is that important? Because I don't want to lose these small details. They're very subtle. And I also don't want to have folding tissue because this is unreal for histology. And also, I need colors. It's not very usual for registration to work with colors, but in our case, histology, different stainings will have different colors and they will carry different meanings. They carry different information. So one of the requirements are having this volume in color. What we did is like the registration only runs on grayscale. So we split the original images in red, green, and blue channels. Those volumes, and they become 3 volumes, and they go through the pipeline, except that I don't need to rerun the registration. I saved all my affine matrices and my deformation fields. I just reapply them to these volumes. I combine them at the end, and I have my color registration. By this time, I end up with around 70 gigabytes of data. That's the size of my data set. And most medical image viewers don't handle well this kind of data. They don't scale well again. Most of them will try to load everything into memory, and they will crash. They'll be very sluggish, and they don't handle well color. So we needed something that where you could see the color and you could be able to work with this big data set, which is not so big for people in big data, but it's big and very big, very large for medical imaging. So we use the ParaView, which is not a medical imaging viewer, but it's great for scalable visualization. So we are able to split our volume in several computing nodes. And this way, you're able to browse through the volume, make videos, screenshots, zoom in, zoom out, zoom to the structures that we want to see. And this is an example of what we get with ParaView. And these are our 3D reconstructions in comparison to our MRI. And here on right, this is my histology, which was downsampled to 0.15 millimeters. And this is my MRI, and I can see that my histology still holds, still carries information from the small inner brain nuclei. This is called the red nuclei, for instance. And this is not visible in the MRI at all. This is another 3D reconstruction done with Paraville. And I'm going to show some of the videos that we made using Paraville. It's the kind of thing that we can do with these volumes. Oops. Yeah. This one is interesting because we can actually here see the inner brain structures, the brainstem. Sorry, the projector is not really helping with the videos. And here, I'm going to show you. So this is the histology, and the red and green structures were actually the nuclei we were interested in. So the second project that I'm working, which is in terms of algorithms unrelated to registration, but we are still working with the same problem, is cell segmentation classification. So basically what do we need to do? We have these 3D structures, and now we need to know what's happening at the cell level. And basically, we need to quantify what's going on with the cells, which cells are dying, which cells have tau tangles, which cells are dying and have tau tangles, because we want to answer the question, are those tangles causing cell death? This is not well known in Alzheimer's yet. And for this project, we were working, I'm going to show you, with images stained for two markers called caspase-6 and CP13. One stands for cell death, cells that are going through a certain cell death pathway, and the other that is stains cells that have the tangles. And we need to quantify these cells, segment them, and quantify in a reliable way. So again, our workflow, we have a brain that goes through the fixation process. This time we don't use the whole brain, you use the brainstem that is embedded in celloid. When the block's ready, it is sliced. So we have some of the slices going through a kind of staining called gallosina, and other slices are going to be re-embedded and re-sliced and stained for what we call immunofluorescence immunohistochemistry. So here they're being stained for those markers that I mentioned, and we are right now working with the fluorescence images. And basically, what do you need to do? We have several images, and the researchers, they have to go to each image, scan the whole image, eyeball the whole image, and count the cells. Yeah, and they have to count the red ones, the green ones, and the yellow ones. The yellow ones are the ones that have overlapping. They are dying, and they have the tangles. And we need to do this in order to find if there is any correlation between the tangles, the cell death, the Alzheimer's stage. We need to do this counting. And this is very tedious. This is very time-consuming. And also, we use human tissue, which gives us a lot of background. We have a lot of debris, like these red structures here. We have a lot of holes like this, the white region here. And this is pretty different from what we see in literature. If you look for methods in literature for segmentation fluorescence, usually you're going to see images from cell culture and animal model, which have a smooth, dark background and very bright cells, so they have a lot of contrast and makes the segmentation much easier. We needed to find a better way to do the segmentation. So a more flexible, smarter way. In this case, I'm working with images whose resolution is about 1.6 micrometers. It's an order of magnitude more than the previous images. So what solution did we find? We worked with something called dictionary learning. And the idea of dictionary learning is, well, from the signal processing field, you can basically represent a signal as a linear combination, either as a linear combination of a basis and a vector of coefficients, or the inner product of inverse of this basis and the signal, and you have the coefficients. Very popular bases, they're also called dictionaries of Fourier, Gabor, and wavelets. And those are called analytic formulations. And they are very popular because their mathematical properties very well understood. They have well understood algorithms, fast and simple. However, sometimes they don't represent well all data. And then comes the fact that you can actually learn a dictionary or transform from the data. And in this case, we use overcomplete dictionaries, meaning that we have more vectors, also called atoms, than the needed, meaning that I have more vectors than the dimension of my problem, the dimension of my image in this case, and my patch. And they're no longer orthonormal. So the idea is that I find, instead of using all the vectors in my basis, I find the ones that best fit my data. So the model is the following. I have my basis D, which is my dictionary, and I'm going to have a sparse representation, and I'm going to only pick the vectors that really matters. They really represent well my data. And I do this by solving an optimization problem, because I no longer can use linear algebra here, pure basic linear algebra here, because I have over the determinate system. It's going to give me infinite results, infinite answers. So I use the optimization, and I have a cost function on the vector A that usually enforces sparsity and enforces that I get the better vectors here. So how do you use this for machine learning? This is used in denoising, this is used in painting, but how do we use this in dictionary learning? It becomes a supervised learning problem. Learn dictionaries from my cells and from my background. What I didn't mention is those images, they were manually counted by other researchers in the lab. So we had this ground truth. And I learned the dictionary by another optimization problem where I optimized the dictionary D and the vector A in an iterative way until I meet some stop criteria. I don't use the A in this case. I'm just interested in the matrix D. And I trained these two vectors, one for background, one for foreground. And the idea is that the reconstruction that uses the right dictionary, the dictionary that belongs to the correct class, the one that uses the smallest error. And so I can use this for classification, and I can train as much dictionaries, as much classes I have. And for my actual classification, for classifying those images, what I did was using sliding window over each pixel. And for each pixel, I would encode the pixel using both dictionaries, and I would have two new patches. And then I computed the error between. I knew I have the original image, which in this case is a patch. Let's say, transform back the encoded patch to image space, and I compare both of them. That's how I compute the error. And my pixel belongs to either background or foreground, depending on the smallest error. This is the mask. I can build a mask using this technique. And this is a mask that I actually built from this image. And I can post-process this mask. And I can use this mask to find my cells. And I can later refine my mask and locate the cells and then classify the cells. And the idea is that we're using this technique to try to build a tool that will improve the productivity of the researchers. So it's not a 100% automated tool. We want a tool that we can guide the researchers to where are the cells and not debris, basically. So our goal is basically understand what's going on in this nuclei, having these tools helping the researchers doing studying Alzheimer, quantifying the tau. And we want to be able to locate this nuclei in the MRI, and we need for this, if we use clinical standard MRI, we're going to need the histology. And so we want to be able to also align well the histology to the MRI, and we want to be able to also use my full resolution histology. So our goals is, one of our ambitions is to be able to scale the registration in the future. What else? We can also, as a let's say, side effect, we can use this registered histology volumes to build histology templates, high-resolution histology templates, which are not quite easy to find around for brains. And basically, any questions? That's like, both of these projects are ongoing projects, so there's a lot of things to be done yet. So,
thank you, Maryana.
Thank you.